Traction force microscopy is used to measure cell-generated forces. This protocol simplifies the collection and analysis of traction force data in order to make it more accessible to inexperienced users.
The main advantage of this technique over existing reference-free traction force microscopy platforms is that multiphoton lithography allows a quick and easy patterning setting redesign according to individual experimental needs. There are many key steps in the fabrication and implementation of this new reference-free traction platform that are easier to understand through visual representation rather than text alone.
For photopolymerization of the base hydrogel, first add 3 microliters of the pre-polymer solution onto a thin flat sheet of perfluoroalkoxy alkanes, and place flat 150 micrometer thick PDMS strips surrounding but not in contact with the drop of pre-polymer solution. Place an acrylate siliconized cover slip on the PDMS with the pre-polymer droplet centered under the cover slip to flatten the pre-polymer droplet to the thickness of the PDMS spacers. Expose the sandwiched pre-polymer solution to UV light for about 1 minute. When a hydrogel is fully formed, carefully separate the cover slip from the PDMS spacers. Use high-performance double-sided acrylic adhesive to attach an open-bottom petri dish to the cover slip.
Take special care when adhering the hydrogel-laden cover slip to the petri dish. A wet dish or too little pressure during the application will allow leaks to form, ruining the sample.
Apply pressure to the adhesive contact surface to create a complete seal between the cover slip, adhesive, and petri dish, taking care not to crack the glass. Use sterile filtered PBS to rinse the hydrogel. Then add 8 microliters of NVP to 800 microliters of the lab solution prepared for the hydrogel synthesis.